Okay, hi everybody, my name is Cass Fields. I'm the Vice President of External Affairs at Intrabio, and I'm going to be presenting with you. You got not a last minute swap, but the name on your program is different, so don't be confused by it. I'm just gonna give a brief update on our development of N-acetyl-L-leucine, which we very simply call IB1001 for the treatment of AT. So this is a fun new compliance slide I'm required to read to you verbatim. So N-acetyl-L-leucine is an approved therapy in the United States indicated for the treatment of neurological manifestations of Niemann-Pick type C in adults and pediatric patients weighing 15 kg. IB1001 is an investigational therapy for AT and has not received UK MHRA approval for any indication. For all indications besides NPC, IB1001 is an investigational medicinal product and is not approved for marketing or commercial sale, and its safety or effectiveness have not been established. All right. We've checked that box off, now let's get into it. Okay, so for some background, the compound N-acetyl-L-leucine, it's a chemically modified amino acid that is the active enantiomer of the racemic mixture N-acetyl-D-L-leucine. So the D-L-leucine, or as we always like to simplify it, D-L-leucine, think of it as this. You separate it into D and you separate it into L. The DL form is approved in France. It was approved in the 1950s for the treatment of acute vertigo. It goes by the trade name Tanganol, but it has not been approved anywhere else in the world for anything else. Our team for the past 15 years has been working on this compound, and when you separate the D and the L, they identified that the L compound, the L enantiomer, is the active enantiomer with both a superior safety profile and efficacy when administered independently. So we have to think of them as essentially three distinct drugs that do different things in the body. But based on all of our research, the L form is the superior one, so that is what we have done in our development program. I just clicked to slide here. It's here. Okay, so N-acetyl-L-leucine. As I mentioned, we call it IB1001. It's just easier. It's an orally administered agent, so it's a powder that you dissolve into a liquid, and then it can be administered either orally or via G-tube. We know that the acetylation is a huge aspect of its pharmacological activity, and without that chemical modification, L-leucine alone would have no therapeutic effect. The drug is taken, as I mentioned, as granules in a sachet that you swallow, and this novel transport mechanism enabled by the acetylation allows it to be delivered to all major organs as well as across the blood-brain barrier. All of these together we have seen accumulate to rapid symptomatic benefits, but also long-term neuroprotective effects. So what are those benefits? Again, as I will mention in AT, this is investigational, be inappropriate to comment on the efficacy, but we have seen a potential to improve a very wide range of neurological signs and symptoms and neurodegenerative disorders. So it was approved in the US for NPC, which is it's also a rare autosomal recessive disorder, and one of the hallmark symptoms is ataxia. So in that disease, we saw a pretty rapid benefit in things like walking, balance, fine motor skills, speech, swallowing, cognition. And that brings us to the development for AT. So during our development program, we had begun with 3 phase 2b clinical trials that did not have a placebo arm. Very quickly, it became apparent, and by apparent, I mean the regulatory agencies told us point blank if there was no placebo, we couldn't proceed with submitting to the FDA or EMA for regulatory approval. So we shifted paths and we created this study design. This is the exact same study design that we ran for Niemann-Pick type C that led to the approval. So we know it is something the regulatory agencies are happy with and that there's a pathway towards success. So essentially what it is you've got a baseline period where you're not on treatment. All the patients will go in for a 12-week treatment, and they will be randomized either to placebo or to IB1001. After those 12 weeks, they cross over and will receive the other. So all patients in the trial will receive both placebo and IB1001 at one point. So in that sense, they can act as their own kind of placebo arm. Then we roll into, if they would like, an extension phase where we're able to collect some longer term data. So recruitment, we began recruitment on May 2nd. As of June 2nd, we completed recruitment, and we have kept recruitment open this month. We're still in June. Sorry, it's very, very new. So we are now powered at about 170% over recruitment, which is fantastic. These are our trial sites across Europe, UK, and the United States, some of whom are here. It's been very nice to meet them. But yeah, so recruitment is now completed, and we expect top-line data in realistically January of 2026. And that just brings me to my contacts page. So if you would like to get in touch, please do. If not, I've listed the details of the NCT trial number so that you can get more information if you would like. But I am excited to be here next time with you, hopefully sharing some exciting data. So thank you.